Okay, thank you for the introduction. So today I'm presenting on behalf of Dr. Shaun Purcell, who sends his apologies from the ski slopes of Colorado. Unfortunately, changes to his flight schedule meant that he wasn't able to come back for this session today. So today I'll give a brief overview of our tool Luna, which was developed in a close collaboration with the NSRR. So the original motivation for Luna was to meet the need to process large numbers of NSRR recordings, with a focus on PSG-derived EEG signals, driven by Shaun's first foray into the sleep world with his 2017 paper on sleep spindles. At the time, although there were some powerful general-purpose tools such as EEG Lab, these weren't necessarily designed to tackle specifics of sleep EEG. Or to process thousands of individuals. So Luna is fundamentally a command line power tool designed to work best in a high-performance computing environment. The focus on scaling automated and reproducible large-scale analysis more naturally led to scripted rather than interactive workflows. And two other design requirements were, first, to facilitate working across multiple, sometimes heterogeneous cohorts, and second, to be able to handle rich annotations and metadata alongside physiologic signals. Since then, Luna was expanded in various ways, adding a range of new functionality. Current developments include extensions to high-density EEG with other signal modalities in the pipeline. As we'll show later, we also sought to make Luna more accessible and added support for more interactive workflows, most recently through integration in the Python-based notebook environment. And finally, we are pursuing tighter coupling of data and tools to support various forms of model-based predictive analysis. And I'll also talk about that a bit later. So we very briefly will step through 6 domains of function, each of which comprises of multiple commands in Luna to give a general sense of Luna's scope and focus. So first, data wrangling and cleaning. Typically, much of the time spent doing analysis is trying to get the data in analysis ready state, and Luna provides multiple features to help harmonize different formats and sources of data. For example, NSRR data often have multiple labels for the same signal or annotation, or signals with different units, reference schemes, and so on. And Luna provides some programmatic ways to alias and remap these terms efficiently. You can slice and dice recordings quite flexibly. So here's an example of Luna script commands to select only certain parts of the recording for analysis. Also, as well as adding, copying, and dropping signals, there are other standards options for cleaning up signals, including resampling, filtering, and others, denoising, artifact rejections options, illustrated here, spectral interpolation to attenuate line noise harmonics. Second, there are commands to quantify multiple Hypnogram metrics, possibly allowing for truncated recordings to estimate non-REM cycles and annotate signals based on Hypnogram-derived measures. If staging data do not exist, the POPs module can generate automated probabilistic staging calls, as shown here, one example. It is quite flexible and appears to perform comparably to other current best-in-class stagers. If staging data do exist, the SOAP command can empirically evaluate the consistency of the staging against one or more signals, potentially identifying some bad staging data, for example, if the file was saved with the wrong name or was corrupted. It does this by fitting a simple model to the data and seeing how well it can recapitulate the observed staging epoch by epoch. And the underlying SOAP model has been extended in various ways to manipulate existing data. For example, it can fill in missing stages, convert manual staging from, say, 20 seconds epoch to 30 seconds epoch durations, or even align staging and signal data if timestamps have been lost. Next, Luna supports a variety of standard spectral methods, including Welch and multi-taper power estimation, the composition of periodic and aperiodic spectral components via ERASA method, the estimation of spectral slopes and intercepts, and other approaches. Sleep-specific functions include spindle and slow oscillation detection, and we've used this extensively in multiple papers. Also, importantly, these can be flexibly parameterized, for example, detecting so-called low amplitude spindles across an arbitrary range of frequencies, as Shaun did in his 2017 paper on spindles. Outside of NSRR, we've been working with high-density sleep EEG and have added support for multi-channel QCs and channel interpolation that operates on the epoch-by-epoch basis rather than reinterpolating entire channels. There's also a range of connectivity measures, including coherence and phase slope index, as well as implementations of ICA and microstate analysis suitable for high-density EEG data. All of these Luna functions can generate a lot of output, stratified potentially by frequency, channel, sleep stage, and so on. And Luna provides a couple of functions to help with high-dimensional outputs, either through dimension reduction or by empirically accounting for multiplicity and clustering metrics at the point of association testing. In the first approach shown here, the so-called principal spectral components function provides a wrapper around the singular vector decomposition, allowing for stratified Luna outputs to automatically assembled into a uniform matrix for analysis, and will project new observations into that PC subspace. As in this paper, we've used this approach to reduce individual differences in power spectra for a single EEG, providing a data-driven alternative to classic band power summaries with a structure that basically replicated across different samples. An advantage of this approach is that it naturally extends to more complex scenarios, including stage-stratified metrics or multiple channels and so on. For example, as illustrated here, where principal component captures variation in spectral power across channels and frequencies during N2 sleep. Finally, we are increasingly focusing on adding empirically derived and model-based functions to Luna. For example, in collaboration with Dr. Sun and Dr. Westover, we've implemented models to predict so-called brain age from the sleep EEG, and we've extended this work to pediatric samples as well. From a tools perspective, we are aiming to make all these models effectively a single-step procedures, going from a raw EEG as input directly to the prediction as output. And we made it relatively easy for other investigators to use this framework to instantiate other predictive models. For example, in our own work, we found that non-REM sleep EEG can be highly predictive of diagnostic status for schizophrenia based on multiple features, including spindle, spectral power, and connectivity metrics. And in the future, we aim to distribute a growing list of these types of model-based predictions alongside Luna's core self-contained analytic functionality. And as mentioned, we are actively trying to make these tools and resources more widely accessible. One effort in this regard is a cloud-hosted web app built on top of Luna called Moonlight. And it provides basic visualization and interactive analysis for individual recordings. We also recently added Moonbeam extension to Moonlight, which lets NSRR users pull individual recordings, annotations, and clinical covariate data into Moonlight for viewing and interactive analysis. A prototype of this was developed by Senthil Palanivalu, and it's available at this URL. A second web app is the recently developed Hypnoscope. It can estimate hypnogram statistics from a single individual staging data, but also runs in a multi-sample mode. Basically, it means displaying hundreds or more individual hypnograms jointly. Users can temporarily align these studies in different ways, for example, by clock time or lapse time, and sort rows to explore properties of a cohort that might not otherwise be obvious. So, for example, here we see children from the chat study. They have uniform clock time at which the recording ends, which may induce spurious correlations between chronotype and sleep duration. And as a second example, in the Cleveland family study, participants were woken for blood draw at a certain point in the evening, and you can see that by this gray band. And depending on the question being asked, these types of factors may be important, and Hypnoscope helps to identify them. Very recently, we released our first Python interface of Luna to supplement our prior R interface. We've created a Docker container, which you can loosely think of as a type of virtual machine that runs on any platform and bundles Luna with the new Python interface, as well as a set of models and resources, for example, for automated staging or biological age prediction that I mentioned before. Whereas for large cohorts and analysis of large cohorts, a command line approach is likely to be most powerful. We can see this Python environment as evolving to a natural front end for Luna, especially given its popularity in the data science arena. The Python wrapper directly binds the same underlying C++ engine as the command line version, and also provides convenient functions to move Python objects in and out of Luna representation of the EDF and its associated annotations. So with that, I hope that I've given a sense of Luna primary aims and scope. Some of these newer parts are still under development, and it certainly has some caveats, but the core library is quite mature, however, and there is a lot of documentation available on the website, including tutorials and vignettes. The Python interface is more recently posted, but interested parties are encouraged to try it out and provide their feedback to us. And we expect the full documentation for the Python component to be posted within the next couple of weeks. And also, please contact us via email if you have any questions. We will be happy to assist when we can. Thank you.
Thank you, Dr. Kozhemiako. We have one question for you right now. Can you talk a little bit about the relationship between purported relationship between deviation from the brain age and chronological age in terms of predicting health?
Yeah, so that's what I can say about this, that this is in general the new framework. It was exploited before using other modalities, including structural imaging or functional connectivity. And turns out, based on sleep signals, we can predict brain age with very high accuracy comparable to those modalities or even higher. And this deviation of prediction compared to observed age actually can tell us something about some pathological processes that happen. So in our own work on pediatric sample, we found that there were some, let's say, negative error or that can be interpreted as a delayed development for patients with Down syndrome. But there are also evidence for that same increased gaps, brain age gaps for other cohorts like schizophrenia. And definitely there is something there telling us about general patterns of aging when we estimate